So hello, sorry for the delay. We had some technical difficulties. I have no idea if everybody can hear me. Do you know, Carlos?
I can hear you quite well.
I can hear you, okay. So welcome Paulo, George and Holger. Robert got here some delay with his with logging in on his computer. So I would suggest that we start. I will talk about the discussion, as we'll start the discussion about how to collaborate within ERNICA and between members, and how we can progress in our scientific endeavors. And Paulo is going to talk about Chabal syndrome, if I'm correct, and George and Ulrich about metabolomics and the microbiome, I think.
Thank you, Aaron. Just a note to all attendees. We will start with the presentations, 3 presentations in all. And after that, we will have time for discussion. If you have a question, you can raise your hand. There's a hand hopefully visible in the panel. Once you click it, then we can see that you want to ask a question, or you can ask a question via the chat, which hopefully is also visible on your panel. So let's start with with the webinar, and I hope you appreciate and enjoy what will be told in the webinar. Erwin?
Yeah, okay.
I will start sharing your screen.
Can you see it now?
I can see your screen quite clear.
The full screen, okay. So, so I would like to open the discussion on what I just said about how ERNICA can aid in and support in collaborations in translational science. So what I would like to present or demonstrate is how genetics has evolved from studying a single gene into genomics, and how it over the years has transformed to a data science. These advancements to the data science holds great promise, but it also raises a lot of challenges about data structures, but it has tremendous potential for artificial intelligence in interpreting the genomic data. So we should consider the storage, organizations of vast amounts of data, as well as the types of samples we collect, how we extract these samples, and how we share these biospecimens and data. So I also would like to introduce you to, maybe you already know, to several consortia that are exploring the same issues and how to solve the challenges that are raised in sharing and working with these large data sets. These consortia are, for instance, the European Rare Disease Research Coordination and Support Action, and the Global Alliance for Genomics and Health. And in the end, I will hope to have convinced you that combining these large data sets has a lot of potential, and sharing data and sharing biospecimens also, but it also needs us to think about how we collaborate and how we set aside our differences. So I would like to start with how a patient comes into the clinic, and then how the genetic workup is. So when a patient comes in, there's a detailed phenotyping and registration. Samples are taken and samples are stored in a biobank, and then genetic analysis is performed, for instance, with whole genome sequencing of its SNP array. And if a patient is lucky, then a genetic diagnosis follows. And that is often when a change in the gene that's already been associated to disease is found. If not, then so if change in the gene is found, but it's not in a disease associated with a genetic change, then functional follow-up is needed. And that nowadays has been done quite extensively in the diagnostic clinic, but most often this has been done is done in a research setting. So the main bottleneck in analyzing and interpreting genetic results, especially for instance whole genome sequencing, is that we can really accurately find genetic mutations and say what they could do, but we are not there yet to definitely say what they will do. And we have a lot of problems with reliably stating what a variant will do if it's located outside of the coding region. So if we find such a variant or a variant in the coding region of unknown significance, then we will follow up with functional experiments to prove the consequence of the genetic change. And we will perform, for instance, animal or in vitro studies. So as I said, it's been mostly done in, I don't know if you can see my mouse, in functional genomics in a research setting. So if you find such a variant, you can prove the causal consequence of that variant in the gene. You want to know if that actually has been seen in more patients, because that strengthens your evidence. So this whole field, the functional genomics and animal modeling, has really made some tremendous progress. So we, for instance, now regularly use gene expression profiling. We investigate protein functions. We added genes for in vitro experiments with CRISPR-Cas. We use zebrafish, or mouse, in which we knock out genes to prove the function and the association to the disease. So all these models are critical for enhancing our knowledge of the genetics and how we connect the genetic variant to the disease. And as you can appreciate in the graph, actually this doesn't stop at the patient. So we have in the end, we find it in more patients, we have samples stored, we screen more samples. And in the end, after a long period of research, we hope to have identified a new disease gene. And actually, then it's been taken up in often in the candidate gene panel, either bioinformatically or in a technical candidate gene panel for screening, and it's been taken into the clinical practice. And then patients benefit. But whole genome sequencing and exome sequencing hasn't been around for that long. But we have been gathering samples for quite some years. So for many patients, we have samples stored in our biobanks and phenotypical data registered in the registries. One of the great benefits of having this in our biobanks is that we can actually screen retrospectively all these patients and then more easily combine these data into large cohorts. And actually, if we do this now, then today's newborn patients actually benefit from all the data and samples that we have collected. However, this has not been done that frequently because it is really expensive and it's hard to exchange data and exchange biospecimens. So also emerging is the transition is that we are investigating patients over time. So from birth into the transition into adulthood. And because we have been collecting samples and data for quite a while, we also have registered comorbidities and symptoms that became more obvious later in life. So linking all these data from electronic health records, from phenotype information, from genomic data, we can actually use this to predict in the future if specific comorbidities will occur based on the alterations that are present in these patients. And if we do so now, we can even maybe implement it for the patients that are born today and find preventive strategies before the symptoms arise. It's also now that gene therapy is slowly becoming more established. There are several approved therapies. So if we find a genotype, phenotype association, or drug sensitivity in our patients, we can already start in our historic patient cohort, and we have samples stored, for instance, fibroblasts or otherwise, or lymphoblastoids. We can already start investigating the functional consequence of these variants, the link with the comorbidity, and slowly starts or aim to develop therapies. So we don't need to wait for patients to be born. So we can now, we have established that we need large genomic data sets for genotyping, and it is also really important to really keep meticulous phenotype information. So we can now think how to gather, store, and share this information. So this is a bit of an issue because in the early days, patients were evaluated with, for instance, karyotyping or Sanger sequencing or SNP array, and that is not really that intensive on type for storage, for instance. So that's a few megabases, a few gigabases in size. But today, most of the patient genomes are analyzed using NGS. And in the early days, that also was that much of a problem because we only had a few genomes, exomes, or transcriptomes. But now we have hundreds to thousands of these really large data sets that we have to keep for really long times. And that is well, cramping up our institute's servers and storage space. Well, it is really important that we actually keep this data, or we should discuss how important we think that keeping this type of data is. So because they're important to identify new diseases, and they can give us new biological insights. So just to give a brief overview of how much the resolution has changed, you can see that it's a relative timeline. So you can see on the left the megabase resolution with the karyotyping, and we could zoom in on genes, and we could zoom in on more genes. And nowadays you can actually measure single cell RNA expression data, single cell DNA, copy number data, and mutation data. And we can place this into with spatial transcriptomics and proteomics into the spatial context. So we have information of cell-cell interactions. And we can now also, with one single technique, measure, we can profile really long DNA and RNA sequences. And you can even measure methylation sequencing. So what is this? It's kind of going to look like is that in the end there will be one sequencing technique to sequence all, so it's DNA, RNA, and epigenome. So the big question is, should we wait because it's now really expensive with sequencing our historic cohorts, or should we do it now? Another hot topic is, of course, artificial intelligence. So genomic data with the ACTs and Gs is really structured, really suited for quantitative measurements and data mining, modeling, and validating models. So computational models, they have relied on patient and control sequence data. phenotypic traits, reference data from proteomes, from transcriptomes, and epigenetic data. And then they, with algorithms, they could predict, for instance, if a variant had a specific effect, if it was deleterious, if it had an effect on splicing, and so on. But nowadays, with machine learning and especially deep learning, it can connect the genome, but also the phenome and the medical records. Genomics has really taken a leap. However, connecting the genome, phenome, and medical records really requires standardization in nomenclature, data formats, medical report structures. So it's really promising, but very difficult to organize it. However, there are several consortia working on a lot of standards. Currently, if you had a grant, then one of the things that the funder really wants is that you store your data, or you treat your data FAIR. So FAIR is an abbreviation for findability, accessibility, interoperability, and the reuse of data. So I'm actually trying to do this for our department, and then it's already really difficult to adhere to the first word, the findability, because in the past, not a lot of people really wrote down or registered what they stored, how it was named, what type of data, a data package, what they call it is. So it is really difficult to even find your data, let alone let somebody else access it, or that you can reuse it in various ways. So for this, it's really important that we are going to keep really detailed metadata describing the data. And this is just one issue of many, one challenge that we are facing. So for instance, the ERNICA tries to solve it with providing frameworks and providing templates. So this is a consortium that aims to build competitive networks, implement effective data collection strategies, and also tries to involve patients more extensively. It is an initiative by all ERNs. And it should enhance the diagnosis or treatment of rare diseases in the future. Similarly, you have the Genomic and Health, the Global Alliance for Genomic and Health, and they also provide a framework for instance, for sharing data, but also for how you should standardize your variant files, or how you should set up your documents for data sharing agreements. So it really provides a framework for all these things, similar as ERNICA. And another solution for the large amounts of data that needs to be stored, and whether it's genome, phenome, transcriptome, or whatsoever, is the European Genome Phenome Archive. So it's the genetic data is privacy sensitive as is phenome data, and especially if you start combining data with artificial intelligence, it really has the potential to not only identify individual patients, but also to identify family members and predict items that or diseases or traits for which the data was not meant. This can have really a lot of benefits, but it can also have a lot of downsides. So we really need to be careful how we share data and how we set up these collaborations to share data, and for that the EGA archive can really help. So in genomics, AI-powered tools So if you have everything set up, standardized, you can incorporate everything, then AI can be a really powerful tool. So in genomics, AI-powered tools are being used to improve the accuracy and efficiency of genomic sequencing. But it can also help identify genetic mutations. So even those that are missed by human interpretation and lead to more accurate diagnosis. So they can really help interpret the functional consequence of your data, of your sequencing data, for instance, by predicting the conformational change of a protein because of an amino acid change, the effect of splice, even the variant has an effect on splicing. And quite recently, they found in yeast, they even managed to predict if a genetic change had an effect on the expression level of your RNA. So that's quite new and really promising, because that was the one thing that was really missing. And in addition to the genome, artificial intelligence does a lot in facial recognition, and they are now also using it even with an app on your phone. The doctor can visit the patient, take a photo. The photo is scanned. This app is called Face2Gene, and there's a really large database of patients and children with genetic anomalies and different facial phenotypes, and it can then look into the database and without even sequencing a patient, predict where to look in your DNA, and it is quite accurate. So this is really the combination of genomics and tools like this is really the future for genetic medicine. So building these large data sets costs a lot of money, a lot of effort of a lot of stakeholders. One of the big issues in data sharing is actually how to share the data. Where should I put my data? How should I? What type of transfer agreements do I need? Etc. Etc. That's one part, and the other part is the incentive of sharing data. It costs a lot of time and money. For funders, really want you to share your data publicly, but for privacy reasons, it's really difficult. But also sharing your data and publishing your data, it really touches your personal academic career. It's sometimes difficult to recognize the bigger purpose of what you're doing. So collaboration is necessary to achieve the objective of enhancing health outcomes. You can discover treatment options and facilitate precise diagnosis, but it also involves numerous specialists, professionals, technicians, scientists, and industry, policymakers. So all those people have to work together to achieve the goal of making somebody better, but every individual also has his own personal goals. So it's, I think, one of the biggest challenges is balancing these personal goals and incentives and the incentives for the patients. But I think as a ERN, we can take a leading role in that. So we can all agree that genomics has transformed into an artificial intelligence interpretable data science with its great benefits, but also its big challenges, also on the ethical side. We really need reliable metadata and naming conventions for organizing, managing our data effectively. It's a really long sentence, but it basically means that we need to describe our data better because we cannot find it now often. We should be aware that combining, storing, and sharing of data, but also the biospecimens that can actually be transformed into data again, has legal and ethical implications. So we should really consider how we should do this as an ERN. So maybe we can take we can set up a set of rules or advice or a framework within the ERN to do this. And this is especially true if artificial intelligence is used. Yeah, so we should carefully consider how we register, what we store, and how we describe and exchange this information. You can. I would definitely advise not to do this yourself, but use templates that you can just download from the several consortia web pages. And maybe we should provide some links in the ERNICA website to these templates. So this said, I would like to open a discussion on how we should enhance our collaborations within ERNICA and how ERNICA can help to set up these collaborations. So Carlos, I don't know how you want to proceed with.
Thank you, Aaron, for this nice talk on clinical genetics. Well, because of the time, I think we should make sure and we continue directly to the next speaker. The next speaker will be Paolo De Coppi. He is from the great Ormond Street Hospital, GARS for short, pediatric surgeon, and he will talk about regenerative medicine, tissue engineering, and then specifically also on short bowel disease. Paolo, I will make you share screen.
Thank you, Carlos. I hope you can see my presentation now. Let me just turn. Yeah, I'll just. Okay. Well, thank you for the opportunity. It's nice to be here among friends to discuss a bit what are the possibilities for the future in terms of regeneration of the gut. And thank you very much, Evan, to have introduced so nicely the genomic aspect. So I also would point out how we as surgeon and clinician needs to be really embedded in the research so we can really move forward and translate into patient. I should disclose some of my interest and patent in the area here. So I'm very fortunate to work as a clinician at Great Ormond Street Hospital, and at Great Ormond we have this new building focused on rare diseases, which allow us to do some of the research I will present, and also the translational aspect. And in fact, in the building that you see there on the left, at the top of the building, there's some clean rooms, 8 of those, that allow us to do translational into the patients. So I think we are fully aware that a pediatric surgeon needs many time to have access to tissue, and that's why many pediatric surgeons being involved of engineering tissue and finding different structure that can lead to a best biological substitute. And I will start from the reverse, so from the patient side, to talk about the approach that we have taken in the past to this aspect. So here is Kieran, who is now 24 years old, who was born with a congenital malformation of his trachea that received many operations that failed. So when he was about 12, in 2010, he received an engineered trachea made from a cadaveric trachea seeded with his own stem cells, and this trachea has been with him since and is done well. However, we know that particularly in this field, there are a lot of controversies, and one of the most known controversies is the fact that some trachea were implanted on plastic, non-desodorized, but plastic trachea polymer, and they were not tested in animals and led to 8, unfortunately, patient death. But on the other hand, other regenerative medicine products have led to cure patients. So one example is the cornea. So now if you have a lesion of a cornea of your eye, you would take stem cells from the other eye, expand them, and then substitute the cornea. And that's become the gold standard. Similarly, we have the example of epidermolysis bullosa. So with epidermolysis bullosa, patients have been treated by using a gene correction vector, using a retrovirus, leading to taking biopsy. And this is a patient that's been treated in Germany, taking biopsy from the skin and substituted the entire skin of the body. And this is a dramatic example. But what is the difference between good example and so-called bad example? Is the amount of research that is on the back of those. And we have to recognize that by doing more research, we don't take it longer for patients, but we make it more beneficial and more effective. So we try to get the same approach to create a functional gut. So a long time ago, we set up a series of partners that were fully involved in this. And the idea is to create a functional intestine. But as you know, this is very complex. So the first that we needed to address was to get a vascularized tissue. As you well know, if you don't have a vascularized tissue, you will not be able to create a functional organs. So for the example I showed you before on the trachea, you can use the omentum, and that was my contribution as surgeon, to take the omentum from the abdomen and then wrap it around the trachea. But if you want to create a more vascular tissue like the intestine that you see there, then it's much more difficult. So we started collaborating with Shaheen Rafi over Cornell University. And with Shaheen, we tried to reprogram some of the endothelial cells to be able to grow them and then seed it on the intestine. So essentially what we've done with Shaheen was to create some endothelial cells that are more embryonic-like, and we use a transcription factors called ETV2 to partially reprogram these cells and make them able to grow. And those expanded cells we are seeded subsequently to see how they behavior and if they maintain the endothelial phenotype. So this is just the example that was done, was to use a bioreactor made in silico, and those endothelial cells were seeded to form network. So those networks were perfused with human blood. You will see in a minute, while this human blood perfused through this bioreactor, there's no extravasation and there's no clotting, which is very important, make us thinking that we could create a more complex network. And indeed, that was the case when we tried to seed those cells in desodorized intestine. Here is a rat example. You see the green fluorescence protein on the lower right panel that colonized the whole intestine. And indeed, when we saw the single vessels, we could isolate the cells present in the vessels, so they were able to colonize that. So vascular supply is the first tick, if you want, on the process, but it gets much more complex than that. So to get functional gut, we had to work on other cell type. So one important cell type is the intestinal epithelium. So of course, the intestinal epithelium is now very well characterized thanks to the work of Hans Clevers and others that have really been able to grow stem cells from the crypt of the epithelium, expanding them, and try to grow them for therapies. One of the main problem of doing that is that we don't have a good matrix to grow organoids, because the matrix that is in use, specifically the matrigel, is can lead to, so it's derived from a tumor formation, so we don't know how to use that clinically. So we focus on a project to try to expand these organoids without using any matrix that couldn't be implanted in GMP. And we found out that the best was to use intestine itself. So we deserialized intestine, and we were showing that we could maintain the structure of the intestine. This is how it looks in transparency, but also if you look at the villi and crypt, we maintain all the structure of the intestine. And here is just some large intestine and small intestine from pigs, which get processed in this way. And you can see here, until you really mince in pieces and you create a gel. So those gels are very similar properties to matrigel, not much of a difference, and the rheology show a very similar gel, which looks very promising to culture organoids. And indeed, that was done after focusing on the fact that this extracellular matrix is clustered very similar to other endodermal-derived tissues, for example, the liver or the lungs. And this is very important because we always think that matrices doesn't matter, but it does matter. And we have for years, for example, used surgices or other matrix derived from the intestine to substitute the diaphragm. And this is not good because actually their tissue of origin is very different. And we proved that also in this work. And but using these gels, we were able to culture organoids from different endodermal tissues. So you see here example of the stomach in the upper left. In the lower left is the liver, which is really comparable to matrigel, for example. There's no differences. And on the right panel, you see various growing of intestinal organoids. And those can grow for a long time. We can really try from single cells, and these gels support that growth. So of course you can use this for research, and as a disease modeling. So for example, Giovanni Giobbe, one of the senior fellow here in the lab, and Brendan Jones, one of the pediatric surgeons, worked together during the pandemic to demonstrate if we could make an in vitro model of COVID. So we were among the first to see that the early presentation of COVID was featuring in children as a gastrointestinal symptoms. And those children would be admitted with query appendicitis, and they were treated medically. I'm sure you have seen many of those patients as well. So Giovanni and Brandon, taking lead from this observation, they tried to recreate an in vitro model for COVID infection. And they were able to reverse the polarity of the organoids. So normally the organoids have the polarity inside the lumen. So the secretion and everything appears inside this ball, if you wish. For getting the infection to the organoids, we had to invert the polarity. So the surface of the mucosa is outside, and we were infected with COVID. And to make a long story short, we were able to demonstrate that indeed in pediatric derived organoids, we could see a infection in the organoids, which was different to the adult derived organoids and proportionally to what we see in children. So this system of the organoids and the gel that we develop help us also on developing a disease model in vitro. But what we are really moving forward, and we hope that we'll be able to achieve, is to try to create a different treatment for children with short bowel, and particularly with focus in midgut volvulus. And the pediatrician and pediatric surgeon in the webinar that will immediately understand the rationale of this. So when you have a volvulus and you lose your intestine, you mainly use your small bowel. But as you know, most of these children preserve their large bowel and the colon there. So we have seen over the years that it's very difficult to create a, if you wish, a all prepare intestine engineer in vitro ready to be transplanted. So we focus only on the mucosa, which is the main difference between the small bowel and large bowel. I'm sure Olga and George will present data on how different is their microorganism, but from the structure point of view, they're not so different. The main difference is the mucosa. So we thought, why don't we look at changing the mucosa of the large bowel and getting the small bowel mucosa in the colon that will allow us to engineer, if you wish, an intestine which is functional, and it's much easier than engineering the whole wall of the intestine. So together with Vivian Lee at the Crick Institute, we start taking biopsy from children with short bowel, and those biopsy, we were able to expand them, and this biopsy come from different parts of the intestine, specifically duodenum, jejunum, and ileum. And we are able to expand these organoids in a volume that was such to replace the entire intestine, because these cells are really growing for infinite time. So we focus then on jejunal organoids because we know how important they are to, if we wish to replace the mucosa. And we were able to not only to expand them, but also to functionally differentiate them, showing features of some typically of the jejunum. Then we focus on creating the scaffolding, and we wanted to test the hypothesis that sitting on the small bowel or larger bowel scaffolding, that would not make a difference. And so therefore, we prepared this cell tissue from the small intestine and the large intestine, and those scaffolding were ready to be seeded with the cells. And in fact, first of all, we show that the two scaffolds are not very dissimilar. If you focus just on the right part of the figure, you will see that there's only very little amount of protein which the two sides of the intestine differ. And this is very important because if you want to make the cells seated efficiently in the two scaffold, we need to make sure that they're not so different. And indeed, we use customized bioreactor that we make in the lab to seed the organoids. The organoids needed to be seeded with the fibroblast, and we know how it's important now to help the organoids to differentiate. And without making a very long story, but these data are published, we could observe the formation of villi in the colonic extracellular matrix, and we could see even differentiation to microvilli structure. So this is very important because it means that we could eventually seed small intestinal organoids into the colon. They maintain the signature. So it means that if you're using from the same patient, you see that in the colon, they may be useful for therapy, even in the same patient. So while we know the stem cell can be derived from different tissues before and after birth, they can proliferate. They're important for the treatment, but also for the disease modeling. There are still various associated unsolved problems, and that's why we need to invest in research in this topic. But regenerative medicine is a clinical reality, and I think this will come out also in the discussion. I couldn't be here presenting if it was not for all the people that has work and work currently in the laboratory and all the funding. Thank you very much again for the opportunity.
Thank you, Paolo, for this nice talk and showing us the technical innovation that might find a place in the clinic. Let's go to the next talk, which will be held by George Singer, who is from the Medical University of Graz, in collaboration with Holger Thiel, who is also from the Medical University of Graz. They will also be talking about short bowel syndrome, but their talk will be focused on the microbiome in the intestine. George, I will share your screen.
Go ahead. Mm-hmm. Does it work?
Yes.
Great. So thank you also from my side. Welcome to this interesting ERNICA seminar, Translational Research. My name, as said already, is George Singer from the Department of Pediatric and Adolescent Surgery at the Medical University here in Graz in Austria. And we here as a group are especially interested in the microbiome. So the first question is, does the gastrointestinal microbiome really matter to us as pediatric surgeons? So we know that in the last, and you certainly all know that in the last decade, examinations of the microbiome have gained an ever-increasing importance in medicine and certainly also in pediatric medicine as well as pediatric surgery. In diseases and malformations relevant to pediatric surgery, such as necrotizing enterocolitis, but also short bowel syndrome, esophageal atresia, and CDH, examinations of the microbiome have been carried out and found really interesting results. So we know that the number of bacteria in our body is actually of the same order as the number of human cells. And especially the gastrointestinal microbiome is a really complex ecosystem that populates the gut and includes bacteria, viruses, fungi, and archaea. The human microbiome develops from day one of life, and to be precise, colonization begins already with birth, and we got colonized, for instance, with a different set of bacteria depending on the mode of delivery, i.e. vaginally versus C-section. The next interesting fact is that the distribution of our microbes significantly differs along the gastrointestinal tract. The fecal microbiome, therefore, does not necessarily reflect the situation in the small bowel. And this fact makes bench work in the lab very, very important, since in the normal clinical situation, the small intestine is not accessible to us as pediatric surgeons. And also the composition of the microbiome depends on a great variety of different factors and can be influenced by, for instance, diet, but also antibiotics and dietary supplements such as pre, pro, or postbiotics. So our microbiome really serves essential physiological functions to maintain gastrointestinal health. So this interface between the gastrointestinal microbiota and the hosts in the gastrointestinal tract is really extremely complex, as it serves several functions at the same time. There is a metabolic role, for instance, and here short-chain fatty acids play a very important role. Acetate, propionate, and butyrate are the three most common short-chain fatty acids, and they are derived from intestinal microbial fermentation of normally indigestible foods. They have a great variety of different biological functions, serving, for instance, as energy sources for colonocytes. They play a role in vitamin metabolism, affect appetite, and in adults, it has also been shown that they can lower blood pressure, making them crucial for gastrointestinal health in total. But they also have a barrier role that prevents the penetration of harmful organisms and the entry of luminal antigens into the host. Regarding the defensive role of our microbiomes, they are very, very important in the development of the gut-associated lymphatic tissue in the pia plugs. And finally, the microbiome also influences mucus production, enzymatic activity, and enterocyte turnover for structural integrity of the gut barrier. So again, I'd like to turn your attention now to the metabolic role of the microbiome, as this is of great importance and in a devastating disease relevant for pediatric surgery, namely short bowel syndrome. As you all know, short bowel syndrome is a rare disease. It occurs with an incidence of about 43 per 1,000 admissions in premature infants. And different definitions of short bowel syndrome have been proposed. One of them is a small bowel length of 100 to 100 centimeter with a colon still in place, or 50 centimeters of small bowel with a colon. Another more pathophysiological centered definition is an inadequate functional bowel, regardless of the bowel length, in the setting of a normal fluid and nutrition intake, making the patient dependent on parenteral nutrition. Treatment goal is always to reduce the rate of parenteral nutrition and/or ultimately to achieve enteral autonomy. This can be achieved either medically with teduglutide, for instance, as a GLP-2 analog, or surgically with the different procedures, or ultimately a small bowel transplantation. The body reacts to short bowel syndrome with adaptation, which is defined as a structural and functional changes, all aiming at increasing the absorptive capacity of the intestine. But one of the main problems that is associated with short bowel syndrome is small intestinal bacterial overgrowth, SIBO, which is defined as a dysbiosis with increased numbers of firmicutes, which are the bad guys, and lower number of bacteroidetes. Factors that contribute to the development of SIBO are, for instance, altered intestinal motility, anti-motility agents, but also acid-suppressing drugs. And the dysbiosis that is associated with SIBO causes abdominal discomfort, diarrhea, and recurring bacterial infections, sepsis, ultimately ending up in a failure to thrive with delayed weaning from parenteral nutrition. However, short bowel syndrome is not only a disease that is limited to the gastrointestinal tract. I would like to turn your attention to the gut-brain axis, which is a connection between the gastrointestinal microbiota and the functions of the brain. And for this axis, there's only limited information in short bowel syndrome SIBO patients. And here in Graz, we have an internationally recognized group that focuses on this connection. There are multiple communication pathways along the gut-brain axis, which partially function bilaterally. These include the gut microbiota-derived molecules, immune mediators, gut hormones, vagal efferent neurons, and spinal efferent neurons. It is important to note that these circulation-based endocrine and neural communication routes do not operate in isolation, but they all are really closely linked to each other. Microbiome metabolites, microbe-associated molecular patterns, they can act both on the gastrointestinal endocrine and the immune cells, and also the sensory neurons. An example are again the before mentioned short-chain fatty acids, which are multi-target messengers that can act on the gastrointestinal endocrine, mucosal, and immune cells, but also on the brain microglia. These interrelations and the consequences are of great interest for future experimental as well as clinical studies in the setting of short bowel syndrome. So to facilitate experimental studies for short bowel syndrome, animal models are crucial. And therefore, recently, we were able to establish a murine model of short bowel syndrome in our lab. We performed a 50% resection of small bowel. We were able to achieve 85% survival in our experimental group at 4 weeks. And from week 2 on, we see a significantly lower body weight of the animals. And additionally, we found significant differences in the fecal microbiome in short bowel syndrome animals compared to the sham operated controls. And these differences were not only found in fecal samples, but also in the different other gastrointestinal sections. So this method really opens doors for future interesting projects, and surely we are open for international collaborations. So these alterations of the microbiome have already been described in short bowel syndrome patients in a number of different studies. Here, for instance, from 2017, showing that short bowel syndrome patients, and particularly those children with poor growth, have a marked dysbiosis. The microbiota differed significantly between short bowel syndrome patients and controls. And there was a persistence of pathobionts and the depletion of beneficial microbiota in short bowel syndrome patients. However, all these interesting studies have only included a limited number of patients. You can see here 11 patients, 7 healthy siblings, 15 patients, 3 healthy controls, and the most recent one published in 2001 in Gut Microbes included 19 SBS patients compared to 37 controls. So In summary, concerning short bowel syndrome, we can say that we know that short bowel syndrome really causes severe alterations of the gastrointestinal microbiome in both the experimental setting and in children suffering from short bowel syndrome. However, the studies have included limited number of patients with different inclusion criteria, different definitions of short bowel syndrome, as well as different underlying causes. And the effect of treatment options remains largely unknown at the moment. Another interesting ERNICA, as well as pediatric surgical relevant diseases, is esophageal atresia. And at one of the last ERNICA meetings, a patient representative stated that for esophageal atresia patients, there are two main things in the long term: eating and breathing. And regarding breathing, the importance of the respiratory long-term outcome of patients with esophageal atresia has been increasingly recognized in the latest years. We all know the children following esophageal atresia repair frequently suffer from respiratory symptoms. And the common opinion that our lungs are sterile has been refuted in the several studies in recent years. And these studies have identified, for instance, up to 2,000 bacterial genomes per square centimeter lung tissue. So, nevertheless, the pulmonary microbiome of esophageal atresia remained unknown. This led us to perform this study, which we were able to publish in 2021, assessing the cardiorespiratory performance and the airway microbiome in esophageal atresia patients in a single-centered study. What we did is we did airway microbiome sampling by deep-induced sputum. We analyzed the sputum using 16S amplicon-based sequencing. We were able to include 19 patients and compare our findings to 19 age and sex-matched controls. Mean age of our patients was 24.7 years. And regarding the microbiome, the main findings of our study were that even though the alpha and beta diversity was not different between the groups, we were able to find bacteria that were significantly different between the two groups. And these were Prevotella, Mogibacterium timidum, and Streptococcus aeruginosus. All three of them are normal commensal inhabitants of the oral cavity. However, Prevotella and Streptococcus aeruginosus have been shown in the pulmonary microbiome in chronic lung diseases, and Prevotella is also associated with an inflammatory response, protracted bacterial bronchitis, and a higher risk of progressing COPD in adult patients. So, to summarize the findings for esophageal atresia, we know that the pulmonary microbiome is altered in patients following repair of esophageal atresia. However, one of the biggest limitations of this study is the relatively small sample size, but larger sample sizes in orphan pediatric diseases are really difficult to achieve. And this is where ERNICA can step in. And the question is, what can ERNICA do? Or let me just rather rephrase this question to what can we do for ERNICA and ultimately for our patients, and not just our patients, but all of the affected patients and their families in the whole of Europe. ERNICA, with its 40 specialized hospitals in 12 European countries, all of the full members, the affiliated partners, but also the patient representatives, really can provide two things. On the one hand, it can provide contact to other institutions and researchers and facilitate us to deepen and broaden the knowledge of both experimental and clinical research. So we really are able to join forces to answer many questions that are still open. And the other thing is we can increase patient numbers of our studies, including patients with the rare malformations spanning the whole of Europe. And certainly we have to include the views and needs of the patients as well as their parents. And this is why we want to start and propose an ERNICA-wide study planned as a clinical observation, including a larger number of short bowel syndrome patients. And we want to have a look at the gastrointestinal microbiome and the metabolome and the nutrition of those patients. So if you want to participate, you're more than happy to welcome to contact me. And with this, I'd like to stop and thank you very much for your attention.
Thank you very much, George, for your nice talk. I will show my screen. So I want to thank all the speakers for the nice presentation, showing nice examples how the lab, the bench can and should connect to the clinic, and with the latest findings and future developments with potential for the clinic. Erin, are you still there?
I am. Can you? I'm here. So we had some really nice talks with a lot of overlap. And one of the goals of this webinar is to create scientific collaborations. I would first like to start if there are any questions from the audience for the presenters. Carlos, how do they?
I still don't see questions yet. Oh, there's a hand from Mario Constantini. Trying to, there you go, Mario. Do you have a question?
Do you hear me? Do you hear me?
I hear you quite well.
Okay, thanks. Thank you very much for every lecture. I enjoyed especially Paolo De Coppi and Georg Singer lecture since I'm a surgeon. The question is to Georg Singer about the microbiome in pneumonia on the lung of the patient with atresia. Do you think is there any relationship between reflux of gastric contents and the microbiome, or on the opposite, with the PPI therapy that often is a long time in these patients? I mean, I know that your series is very small and you don't have any, but I think it's could be interesting if this biome is related to reflux or therapy. What do you think?
I mean, as you said, one of the major issues of our publication is that it was a relatively small number of patients, and therefore we were not able to make the two subgroups, PPI therapy and not PPI or without PPIs. But I do think, and we know that from other studies, that PPI make a difference in the pulmonary microbiome, and this would be certainly one question to be answered in future projects with a bigger number of patients. And again, this is, I mean, ERNICA can absolutely, and we all together can initiate such projects. But I do think that reflux would probably have a role and influences the pulmonary microbiome.
Yeah, keyword would be silent aspirations and subclinically chronic infection. And I think we owe it to our patients, and only the huge number of centers in ERNICA can give us enough probes to test that. Beautiful question, and that's exactly the way we want to go.
Okay, thank you, Carlos. I cannot see if there are any questions. I don't see hands or.
You can ask, hello, I was unmuted. You can ask your question by showing your hand or typing your question via the question option in your panel. So if you have a question, you can raise your hand. No questions so far, Erwin?
No, I can't see it. So I can have a question. So one of the goals was collaboration, and a lot of the ERNICA conditions and diseases they vary quite. So you can only take a biopsy once, for instance. Are there any ways, for instance, for short bowel syndrome, that we can reuse the biopsies? So for both your studies, so for studying the microbiome and for making organoids, for instance. And how would you set that up? Maybe a question for George.
If there are biopsies and if they're stored at -80 degrees, then we can certainly use them, yes. The question is how many centers do have biobanks that keep these biopsies, not just at normal histology, but because that really doesn't work, but really putting the biopsies at -80 degrees.
So for instance, we have esophageal biopsies that we take after birth, and then we sequence them. And I don't know if we also sequence the microbiome, for instance, because it's unbiased and it never aligns because I don't look for it, for instance. And could that be an approach to first look in data sets of somebody else if there's microbiome in it?
Sure.
Yeah. Because that's the cheapest option, I think, because it's already there.
Okay, I think I also have another question. If I can see.
Carlos, should we encourage people to attend the next seminar? Can you tell us when that will be?
Then this is the first of a series of the ERNICA research collaboration webinars. And of course, we're going to evaluate, but certainly we will plan, we hopefully plan new webinars on these topics. And of course, once we have planned new ones, then we'll of course update you and keep everybody informed via newsletters and website. I can at least tell that the webinars on the quality of life when it comes to research already have planned a series of webinars for this year. There will also be a webinar, a second webinar organized on clinical trials, probably after the summer. And naturally, of course, with this interesting topic when it comes to translational research, I think it's very interesting to find a second webinar. And I'm gonna check if there are any more questions, because I think there was a question related. Let me check again. No, that question has been taken out. Well, we're talking about, of course, in this case, about translational research, and which is, of course, already making use of multiple disciplinaries, if taken the lab and the clinicians. And you all gave nice talks about the projects, which requires a lot of collaboration. And one of the aims of this webinar, and we'll continue, of course, this is how we can use ERNICA to create more projects and to improve the collaborations. So that was the aim. Hopefully, we can bring together the people who have been listening to the webinar, and with the examples you've given, we hope, well, that in the future, ERNICA might be even stronger position to have more projects on this level. Do you have any suggestions? I'm talking to the speakers, or ideas. You already mentioned a few with the role of ERNICA, and how would you use ERNICA for future projects? It's a broad question.
Well, if I can, so for me, for instance, for data exchange and for sample exchange, it would be really helpful if I knew who has samples, who has data, who wants to collaborate, and now I know for some people, of course, but I don't know if there's a platform for that or if there's a route for that. And if we do collaborate, it would be nice if we have a common and from the start, from taking informed consent onwards, common forms, so you know that you can already exchange data, exchange samples, and that you have consent for that, and that you don't have to ask it again and again in each institute, for instance. But that is one of the things that ERNICA and via ERICA, I think, should help actually.
Yes,
yeah.
Thank you. Do you have? I see no questions from the audience. Do you have something to add to that, Aaron?
No.
We're almost 5 o'clock. And we like to be on time.
Yeah.
Well, in that case, if there are no questions from the audience, then I'd like to thank everybody for your time, and especially want to thank the speakers for giving their presentations, interesting presentations on the topic from bench to bed. I'm really sorry that our moderator wasn't able to join. There was some network connection problems. Of course, problems we will solve for the next one. And once again, this was the first of a series of webinars when it comes to research collaboration. I already mentioned that there will be follow-up webinars on the 2 other topics, and I'm sure that also when it comes to translational research, we will have follow-ups, and all aim to make sure that ERNICA is stronger when it comes to research, and in this case, has a strong position when it comes to translational research for future therapeutic applications. So if there are no more questions from the audience, and if maybe speakers do have something to add to the last comments.
I got a question from Maria Alves via the app, because it's a really difficult tool to use for the congenital short bowel patients. If Maria asks if the microbiome is similar in between all these patients or comparable.
In congenital or in acquired patients, or do you understand that correctly?
I think congenital short bowel. So because you have acquired and congenital, I think in congenital short bowel patients, because they have the same genetic defect, I think. So it's the
again, I think that the numbers of the included patients in the reports we have yet are too low to really answer this question.
Okay.
So I cannot answer this question.
No,
sorry.
You have to wait. Yeah, it's he's now texting. Well, I think she's texting text. That's right. Okay, if there's no more questions, then maybe we can close up. Carlos?
Yes, we can. Because it's almost 5 o'clock and I don't see questions as well. But I think we learned a lot today about the possibilities, and we will continue in the future on this topic. So I want to thank you all again for attending the webinar. I want to thank again the speakers for the nice presentations, and we hope we will see you soon for the next one.
And we would like to thank you for including us. It's quite an honor. Thanks.
Thank you.
You're welcome.
Thank you.
Okay. Bye.
See you next time. Thank you.
Bye-bye. Thank you.